if you want informative classes of pharmacy, chemistry, medical courses, motivational speeches, poems, please go through yours Emdad channel.
Hello everyone, myself Dr. Emdad, and in the second part of the video I will explain the basic procedure for genetic engineering. And what are the steps? First one, DNA extraction. Second one, gene isolation. Third one, modification of the gene, which is also called recombinant DNA technology. Fourth one, inserting of gene within the target organism, which is called transformation, and the final one is conformation. In the first step, what you'll do, you will go for the DNA extraction, from whom you take two organisms. One organism from which you will collect DNA, which is responsible for your required protein or hormone, say as human. Okay, and second one is that specific organism in which you will produce that target gene, say as E. coli. So both of the cell you collect, and after that you go for the DNA extraction from the both of sides separately. It's a simple procedure. And after that you have to go for gene isolation, and specifically from the target organism. That means if you take a human gene, so for the human gene, what you do, I already told in the earlier video that for human, that 11th chromosome is responsible for that formation of your insulin. So what you'll do, that you will collect that specific chromosome, and within that you have to target, you have to collect that which is specific, the portion of gene of that DNA. Okay, so you have taken from that your human DNA, and now you will go for the modification of gene. And for the modification of gene, which is also called recombinant DNA technology, and you can do by mainly two methods. First one, the mostly used is the enzyme method, and second one is by cloning vector. And what are the enzyme? We have to use the first enzyme, which is called restriction endonuclease. Then what is this endonuclease? Endonuclease means that specific enzyme that can cut DNA. Okay, and for that higher organism, you will cut the specific DNA with restriction endonuclease. Restriction means you restrict, and the function of endonuclease is just like a scissor. It cut the specific part of the DNA. Normally this scissor is used to cut your paper, cloth, such like substance, but this endonuclease, they are specifically for cutting the DNA. And one thing more, what is endo and what is exo? Endo means inside of the cell, exo means outside. So now this endonuclease, they are having also, I told, the restriction. That means they will cut specific gene. Now how they cut, and what is that specific portion? They normally prefer the portion is called palindrome. Now what is palindrome? Palindrome, I am saying with a simple example. You take a word madam. Madam, you write M-A-D-A-M. Am I right? If you reverse it, it is also say madam. In a similar way, you will find it out in the DNA sequence. Like you can say C-A-G-A-C. C means cytosine, A means adenine, G means guanine. So this sequence you can easily cut. Before cutting, you should be careful that your specific gene should contain your promoter and terminator portion, because you already know when you go for any protein synthesis for the lower organism, they need operon, and for the operon you need this two specific position, the promoter position, which actually promote for the biosynthesis of specific protein, and the terminator gene is responsible for stop of that function. You know, if it does no break, then there is serious problem. Whatever. Now, if you say, if you consider my these two finger, okay, and this finger, this is that DNA. This is one chain, this is another chain, two chain. Okay, and when this specific restriction endonuclease cut, they cut like this. You can see there is some sticky part, here is also sticky part, and this will help to join on another DNA. Okay, and you can do for, you can do, you should do for both, for plasmid DNA as well as for that higher organism DNA. And you understand how this sticky portion, you just see, if you join two wood together, what do they do? Like this, you cut some portion from this wood and some from this, and they join together. I've seen already similar way the DNA also be joined. And how will join? For the joining you require an enzyme which is called ligase enzyme. Okay, so ligase enzyme, they can beautifully join this two fragment, one of plasmid and another from that human gene. So they join together and make a recombinant transmit which having that gene of higher organism. Okay, now what you have to do, you have to insert this gene to your target like E. coli bacteria. Okay, and you can do very simply by using a stress like thermal or electric shock, and the procedure is called transformation. You know, bacterial DNA is quite permeable. Already I told in the last video that they can easily transmit of one bacteria to another bacteria very easily by this process. So similar fashion, you also incorporate this specific recombinant plasmid within that target bacteria. Okay, now what then do? Then they will go for multiplication. First DNA will replicate, then they multiply to numerous cell. You know that this DNA, they multiply very fast, and after that they will produce specific RNA by the process transcription, and after that they produce protein by the process of translation, and you will get huge amount of your target molecule like insulin or other protein. Now you isolate your required insulin or such other target protein from that bacteria by simple extraction procedure, and purify it and produce the specific dosage formulation like injection or such like for as per your requirement. Okay, now how to confirm it? Because the last step is confirmation. You can confirm by the procedure is called specific PCR technique. Now what is PCR? That is polymerase chain reaction, through which you can go for the checking of this along with southern hybridization or DNA sequencing. In this way you can understand whether your specific gene was incorporated within that your target or organism or not. And what are the other process? Other process are like transfection. Then what is the transfection? In the way of transfection, you can incorporate a foreign DNA to the animal. Okay, and you can do it through the simple technique like liposomal technique, yeah, through microinjection. Okay, so in this way you can easily insert a foreign DNA within the higher organism. And in this one I want to say for this that one term you may heard, that is knockout gene. Then what is knockout gene? Sometime you may require to remove some gene of some specific organism which is not required, and in this procedure of genetic engineering you can do the what are the gene is removed from that specific organism is called knockdown gene. Now what are the other procedures? In the plant you can go through that agarobacterium mediate recombination, and nowadays the most of the crop are made in this way. And what are the other? The process also is transduction, through which you can easily incorporate your required gene within higher organism by RNA containing retrovirus or DNA containing adenovirus. You know, this virus, they have the capacity to infect you. So before incorporating that specific gene to the higher organism, you first remove that specific DNA, yeah, RNA of that specific virus, and that DNA or RNA which is responsible for the virulence. Then what is virulence? Because this part is responsible for infecting higher organism. So if you do this, then you can easily incorporate our DNA to the higher organism. And also I told you can take help of knockdown gene. Now these are the way through which you can easily target a gene wherever you want. And this is a brief procedure of this. And what are the advantage? What are the possibilities? As well as what are the disadvantages? I will explain everything in the next video. Please go for that. Thank you for your patience. And if you have any comments, please mention it. And if you like it, please share to your friends. Thank you.